Good evening, everyone. So it's a real pleasure to welcome you for SAO's inaugural lecture. And today is not really only the recognition of exceptional scientific achievements, but it's also the celebration of her leadership and her contribution to our academic community. So SAO began her academic journey studying human genetics and cell biology, I think, at the University of the Azores, probably one of the most remote universities in Europe. I have to confess that I had to go on ChatGPT and ask whether there was actually a university in the middle of the Atlantic. And yes, ChatGPT reliably tells me that there is 3,000 students in the University of the Azores. After completing her PhD, SAO undertook several postdoctoral positions. So she went in Spain, in the Netherlands, back to Portugal, before finally joining us here at UCL Institute of Neurology. And today she really leads a dynamic research group. She's using genetic, epigenetic, and multiomics to understand the complex biology behind neurodegenerative disorders. But SAO's contribution go far beyond the lab. And in my EDI capacity, I can really say that I've had the privilege to work with SAO a little bit, and I've witnessed not only her professionalism, but also her kindness. She's a passionate advocate for career development and equity in science, and she was recently awarded with the Faculty of Brain Sciences Contribution to EDI in Practice Award. She also holds a prestigious senior fellowship from the Alzheimer's Research UK, and she is a true role model when it comes to securing successful funding. Her contributions span far beyond her own research group, extending into teaching, but also into public engagement, where she also makes a meaningful impact. In short, SAO is not only an exceptional scientist, but she's also an outstanding academic citizen. And you'll be pleased to hear I didn't just interrogate ChatGPT, I also asked her group, and here is what they told me about SAO. It's hard to find the words to say thank you to SAO. We know we are incredibly lucky to have her in our lives. Her consistent support and guidance are part of what gives us the confidence to grow and pursue our dreams. When we face difficulties, we can remember her with a cup of microwave coffee in her hand, reminding us to breathe. Now it's our turn to cheer her on this academic achievement. But we, Anna, Kate, and Naomi, I see them over there, want to emphasize that who she is as a person should be celebrated just as loudly. So I think it's a perfect word to finish. Please join me in welcoming Professor Conceicao Bettencourt for our inaugural lecture.
Thank you so much for your so lovely and kind words, and the words from my own team as well. And thank you to everyone that joined us here to celebrate this moment, this important moment in person, but also to those that are attending online. And today I've titled my talk, Connecting the Dots, a journey through genetics to epigenetics. And I hope I can convey the message by the end of my talk. I couldn't start my talk and talking about my journey without thanking very important people to me, and that is my family. So my parents, for all their support and for all their efforts for me to study. My parents-in-law for adopting me as the middle daughter, but obviously to my husband and my daughter who are here in the audience, who are my closest support ever, even when life is tough on us. So thank you very much. Obviously, the Azores were already mentioned, so I had to say something about it. And indeed, I'm originally from the Azores Islands, located in the middle of the Atlantic Ocean, over there. And there are 9 volcanic islands, some bigger than others. I'm actually from Graciosa Island, where I was born and raised, and it's the second smallest, so with less than 5,000 people living there. My second home was San Miguel Island, where I went to the university to study my biology degree. And here I am at the University of the Azores, in the university campus, at the end of my degree, what would be kind of equivalent to a graduation ceremony, but over there. And I'm wearing there the students' formal costume from the University of the Azores with that hood on, which I think was inspired in a traditional costume called capote capello. That it was a fashion item that women would wear until the early 90s. I studied biology, but I always knew I wanted to work on something related with human health. And so Manuela Lima was the director of the biomedicine center at the University of the Azores. So I went to have a chat with her and about possibilities to do so. And so I did a first internship followed by a research assistant position together with Manuela Lima at the University of the Azores, but together with the Department of Neurology of the local hospital with Dr. Jorge Santos and João Vasconcelos. And so my first tasks were to bring together, organize information, familial information, about families with hereditary ataxias and spastic paraplegias. So I had to go to the parish records and try to decipher this amazing handwriting to try to extend the genealogies of these families, trying to connect smaller families into bigger families. I also went around the island with the neurologists, trying to raise awareness among GP practices about these diseases, which were not so known across them. I asked Copilot to draw me a road trip from Ponta Delgada to Nordeste. And it is a clear example where AI failed what I asked, because we can't go over the ocean by road, right? So, but in the end, I also asked it to go through a couple of other places there. And the overall picture, I thought it had an interesting shape. So kind of a brain, but without the cerebellum being present there, which made sense. I was working with cerebellar ataxias, which have cerebellar atrophy, right? But actually, my first experiences in the wet lab were not working with human health, but with human population genetics. And so I learned a lot about PCRs and other things, but doing markers of Y chromosome and mitochondrial DNA and collecting samples for this study on mitochondrial DNA mutation rate. And I'm very grateful for Rafael Montiel and Cristina Santos, who were of great support at my early days at the University of the Azores in Manuela's group. I also had a chance to contribute towards this study. So it's still population genetics, but it's now looking at a gene that is causing disease, human disease, Machado-Joseph disease. And this work was led by Manuela Lima at the University of the Azores and Patricia Maciel at the University of Minho. And this set the scene for me to go on and do a PhD with both of them. Obviously, on genetics of Machado-Joseph disease. So this disease is highly prevalent in Portugal, but especially in the Azores Islands, where it reaches the highest worldwide prevalence. And so it made sense to study that disease for my PhD being based at the University of the Azores. Initially, it had actually been thought that it had Azorean origin, and why is that? So there were 3 families that were described back in the 1970s with this hereditary ataxia and additional neurological signs in 3 families living in the US, but all of them with origin in the Azores Islands. So they were described as different clinical entities and only later on unified as a single disease. The first family was the Machado family, then the Thomas family, and then the Joseph family. But given their origin in the Azores, at some point it took the name of Azorean disease of the nervous system. It was only some years after when Paulo Coutinho and others described several other families, including a family without Azorean ancestry, known Azorean ancestry. And so they said Azorean disease of the nervous system is not correct. We prefer to call it Machado-Joseph disease because Machado family was the first to be described. And the Josephs represent the largest and most comprehensively described family. So Paulo Coutinho was a Portuguese neurologist, a woman ahead of her time, here depicted in this picture as the only female neurologist at the department at that time. Clearly, there was no Atenas I committee back then. And I had the pleasure to meet her and many other top experts in the field in this international workshop that we organized in the Azores. And I, as a PhD student, had the privilege to contribute to the organizing committee and bringing over people from all over the world, from Japan, Brazil, US, Australia, and many other places around the world. So it was quite an enriching experience for me to be meeting these leading people in the field, but also learning about logistics, having to organize flights and hotel stays and registrations and many other things. Living in islands comes with challenges, not only for us as researchers, but for the patients themselves. So the smaller islands will only have GP practices, not hospitals with specialized clinics. So if patients need to be seen by a neurologist, for example, they would need to fly over to the bigger island to be examined. But I was fortunate to be part of this multidisciplinary team working on Machado-Joseph disease. So together with neurologists, psychologists, clinical geneticists, and researchers, we would fly to the smaller islands in these small planes with the propellers and everything. Just to give you an idea, from São Miguel Island, the biggest one, to Flores Island, it's over an hour flight. So it's not that close. It's not that we can take the ferry and just go. Obviously, my job here was to do research. It was an important moment for me to be able to explain my research to the patients themselves and their families. And I was overwhelmed with their stories and their reactions. I've always said I'm not promising a cure yet, but we are doing our best to understand the disease, so that could be possible one day. And they would all want to participate and say, I know this is not for me, but maybe for the next generation. So Machado-Joseph disease, also known as spinocerebellar ataxia type 3, is part of a larger group of disorders caused by repeat expansions. And that includes a disease well known here in the department, Huntington's disease. So both of these diseases are caused by a CAG repeat expansion in the coding region of the gene, leading to an expansion of a polyglutamine tract in the corresponding protein. So there is a normal range of repeats, And then when it goes above a pathological threshold, then the disease manifests. It is known that there's an inverse relationship between the size of the expanded repeat alleles and the age at onset when the disease manifests, but the repeats themselves don't explain everything. So there must be something else contributing to that variation. So I wanted to know a bit more about the gene that causes, where the mutation that causes Machado-Joseph disease is. We didn't know much about variation at the RNA level, so I compared RNA with DNA in terms of CAG repeats. There's slightly more in the RNA, but one or two more just. But after a lot of cloning, a lot of sequencing, a lot of sequence alignments, which back then had to have some manual input. And there was no long-read sequencing back then. I have described 56 alternative splicing variants for this gene, including 2 novel exons, and 50 out of the 56 were novel transcripts as well. 26 only found in patients. Thanks to this work, we have this long list of transcripts for the ataxin-3 gene in databases such as the Ensemble Genome Browser. As therapeutic approaches are evolving, and I guess we will hear more in Joe's talk, and with the approval of splice switching therapeutic approaches for neurological disorders, information like this can be key to allow development of therapies for these diseases. Not much was known about other things that were modifying the phenotype, the age at onset. So after a paper came out on investigating ApoE in Huntington's disease, we thought it would be a good idea to have a look at Machado-Joseph disease as well. And so what we found was that the ApoE2 allele, in this case, was associated with the increased risk of having the disease earlier. And this study was, the findings from this study was replicated later on. So my PhD finished with my PhD Viva, which in Portugal is a public event, similar to the one we are having here, but we would have some examiners up there. And I would be sitting down here. It all went well, I guess. And I'm very thankful for both my supervisors, Patricia Maciel, but especially my supervisor and friend, Manuela Lima, who was always there to support me. I don't know what I said, but at the end, after those serious moments, they were laughing for some reason. My journey on MJD did not finish there, so even after I moved to other places, I continued collaborating with Manuela, and I was lucky to supervise both Mafalda and Ana Rosa through their PhDs working on different aspects of this disease. I was finishing my PhD and I wanted to expand my experience in neurogenetics. So while writing my thesis, I also wrote my postdoctoral fellowship, first one. And I went to Madrid to work with Justo Garcia de Yebenes, who was a neurologist working at the hospital Ramon y Cajal, taking the name of the famous person in neuroscience. And over there, I not only expanded my neurogenetics expertise to many other neurogenetic disorders, but I also had to improve my Spanish and not just use my Portuñol. I was also fortunate to visit Peter Oyting's and Patrice Ritsu labs in Amsterdam, where I went to learn about next generation sequencing. It was at its early days back then. And using families from Spain and this technology, we were able to identify causal genetic variants for several diseases, including several ataxias and spastic paraplegias. I also went to Santiago de Compostela to learn several other techniques and to have very interesting discussions about genotype-phenotype overlap in neurogenetics with Suzy Sobrido. And we wrote that paper together. The logical next step would be to come to the home of this inspirational woman, an expert in ataxias, spastic paraplegias, and also neurogenetics. So I came to UCL, Queen Square Institute of Neurology. I joined Henry Holden's group in the department led by John Hardy. Obviously, I applied what I had just learned, and we identified more mutations causing neurogenetic disorders. But my research expanded to diseases, not only Mendelian diseases, but more complex and some of them sporadic diseases. I have a non-comprehensive list of diseases there. And I was very fortunate to meet these people, lovely people, that were doing their PhDs and to help with supervision of their PhDs. They are now successful researchers themselves, most of them back to their home countries, Sarah in Germany, in China, Lucy in Argentina, and Stephanie is here in the room, and she's doing really well here at the IoN. I've always been puzzled about links, things that are shared, things that are common. And after the GWAS in Huntington's disease identified DNA repair pathways has a modifier of the age at onset in Huntington's disease, there was this opportunity to see if it could be expanded to other polyglutamine diseases. And so we ran this collaborative work with samples from many places in the world. And we indeed found that DNA repair pathways are a common mechanism, not only with respect with Huntington's disease, but is also applicable to other polyglutamine diseases. And DNA repair is a hot topic here in the department. That gives more importance to this work, because whatever is found in Huntington's disease can potentially be translated to these rarer diseases as well. I'm very thankful to all my joint first authors in this work, but also to the joint senior authors, including Sarah, our head of department. Again, finding links. So cerebellar ataxias, we have a particular vulnerability of the cerebellum, this red region here in all of them. So I wanted to explore ways of trying to understand why. When I joined the department of John Hardy, they had just published this amazing resource with transcriptomic data from over 100 individuals and covering 10 brain regions, including the cerebellum. And so I've used this resource to try to identify links between genes that cause spinocerebellar ataxias. Back then we knew 24. And so we tried to understand what's common, how are they linked, and what are they doing. So we found two cerebellar transcriptomic signatures enriched for the SCAD genes. On one hand, we have here this brownish signature which we found to be associated with protein degradation pathways, and often associated with more complex phenotypes. But we also found this one here that was pretty much very specific of the cerebellum, and it was found to be involved in calcium signaling, which is quite relevant for the cerebellum itself. We could also find links and patterns in terms of the expression of these genes in the brain. While the yellow signature was clearly more expressed in these cells here, the Purkinje cells, the other one was much more expressed in this region here, the granule cells of the cerebellum. And I don't have the time to go over all the details of this study, but basically, after looking at these patterns, at these relationships, we could link a lot of ataxia genes, not only the SCAD genes, into these coalescing pathways that we can see here in this picture. That approach had worked quite well for the ataxias, and so I thought maybe I can apply it to other diseases as well. And so I did for neurodegeneration with brain iron accumulation. And we again found specific transcriptomic signatures, one of them connected to neurons and synaptic transmission, and another one more relevant for oligodendrocytes and myelination. In these diseases, we have brain iron accumulation, as I said. Oligodendrocytes are the cells with the highest levels of iron in the brain. And that's because they have a high demand, metabolic demand to produce myelin. I was fortunate to be able to work along Juan Botilla, who is now a professor of computer science and machine learning in Murcia in Spain, and Liz from Australia, who provided data. She's an iron metabolism expert and provided data for this work. So I told you about DNA variants, genetic variants. I told you about transcriptomics and gene expression, but what's regulating this? So I wanted to know more. So we know that roughly every cell in our body will have the same DNA sequence. There can be exceptions, and I'm aware of them. However, that would not be enough to explain why cells differentiate into different cells that exist in the blood or the very specialized cells that we have in the brain. So they need different fates, and they need dynamic gene expression. So there needs to be something over the DNA explaining this. Here enters epigenetics. So under the influence of internal or external cues, these epigenetic factors will regulate gene expression in this dynamic way that cells need to function properly and to differentiate into different cells. For this, there are multiple epigenetic marks, histone modifications, non-coding RNAs, but my research has been focusing on DNA methylation, so DNA modifications. DNA methylation consists on the addition of a methyl group, a chemical tag to the DNA, without altering the underlying DNA sequence, but allowing this additional layer of instructions for cells to know how to dimmer the gene expression, the way they need. And I was very lucky to join Janice's group. I'm very thankful for that. Based at Queen Square Brain Bank, led by Tom Warner, to study epigenetics of multiple system atrophy. Multiple system atrophy is like Parkinson's disease, is an alpha-synucleinopathy, but instead of having alpha-synuclein accumulating in neurons, as it happens in Parkinson's disease, it's characterized by accumulation of alpha-synuclein in glial cytoplasmic inclusions in oligodendrocytes. So MSA is a very good example where we can see under the microscope something going wrong with oligodendrocytes in a neurodegenerative disease. Along the way, I was also very fortunate to meet and work with Robert, who was an amazing person, very kind, super dedicated to research. And so we did run the first MSA epigenomide association study. And I also worked along Sandrine, who started the lab work for this work, and Emmanuel Vera as well. One of the main findings from this study was a DNA methylation change in a gene called MOPP. MOPP encodes a protein called myelin-associated oligodendrocyte basic protein. So as the name is saying, it's highly important for oligodendrocytes. It's involved in compacting and stabilizing the myelin. And these methylation changes were found not only in brain regions severely affected by the pathology in MSA, but also in brain regions very mildly affected by the disease, meaning that these changes are happening early in disease rather than being just a downstream consequence of the pathology and neurodegeneration. We had already seen in a collaborative work with Ignacio and Matt from the US that this gene is downregulated in MSA, in white matter tissue, as well as in oligodendrocytes. So we looked at the relationship between methylation and gene expression, and methylation is actually driving downregulation of this gene in MSA. We wanted to know more what happens in terms of the protein. So together with my collaborator, Yasumiki, who by then was visiting the Brain Bank and Genesis group, we found that the patterns of the expression of this protein are very different in MSA compared to controls. And we actually found that MOPP is trapped into the pathological inclusions in MSA. We also tried to quantify the protein load. We didn't find any differences between MSA and controls in this case, but that was kind of not expected based on what we saw in the RNA. But given that the protein is getting trapped into the inclusions, it's not being degraded probably. But we also compared with other neurodegenerative diseases, including PSP and Huntington's disease. And it seems that the levels of this protein are actually higher in those diseases. And I worked along with Rohan, who told me a lot of tips about Western blotting, but we also had very interesting discussions about protein isoforms for MOPP and alternative splicing. This gene had already been linked to neurodegeneration through GWAS, including this GWAS, which describes MOPP as one of the risk alleles for PSP and had the contributions from IoN. It has been replicated across studies and in different neurodegenerative diseases, including a recent PSP GWAS. So when Kate came along as the first PhD student in my lab, we thought it would be a good idea to explore this gene further and look into several neurodegenerative diseases. And so she did. She did look into methylation, gene expression, genetic variants. Some of this work was done in collaboration with Kurt and Jack. Some of you may recognize Jack's face. He's a a former UCL student and has been a great collaborator over the years. I won't spoil you too much about the findings from this work, as we are still finalizing some of the analysis. But what I can say is that although this gene is dysregulated in multiple diseases, the mechanisms contributing to that dysregulation might differ across diseases. In some, methylation will have a stronger effect, while in others, genetics will be playing a bigger role. We also looked across Parkinsonian diseases in terms of changes in DNA methylation, and this was work led by Meher Murthy, a postdoc in the lab. And we found quite a lot of similarities, despite these diseases being different. Molecularly, they do share dysregulated DNA methylation signatures. But we also saw that MSA and Parkinson's disease, which are both alpha-synucleinopathies, shared more than they would share with PSP, which is a tauopathy. Dementia affects so many people in the world, in the UK, and my own family. And being inspired by the work of Tammaryn, Lashley, and John Hardy, I also wanted to contribute to this field. And so I went to them and said, I want to apply for a fellowship. Would you support me on this? And they both did. And actually, when I went to John, he said, if there's someone that can help you with this, it's Tammaryn, and she has been giving me great support in establishing my own lab. So I was very fortunate to have them both as sponsors of my two fellowships, a junior fellowship and more recently a senior fellowship, funded by Alzheimer's Research UK, and I can see someone representing them here in the room. Thank you very much. But first, talking about frontotemporal dementias and their pathological forms, which take the name of frontotemporal lobar degeneration. It's quite complex. It includes different clinical phenotypes, different pathologies with tau pathology, TDP43, and others. It includes genetic forms of disease. It includes sporadic forms of disease. But we do know they all have shared selectivity of the frontal and temporal lobes of the brain. They must have something in common. So in this work, led by Kate, who I mentioned before, we looked at what could be shared across different subtypes of FTLD, including TDP and tau pathology, looking at brain tissue, post-mortem brain tissue, looking at different data sets that would comprise sporadic and genetic forms of the disease to try to identify shared dysregulated signatures and loci across them. We were lucky to collaborate with several people in this work and others as well. One of the main findings was methylation change in a gene called OTUD4. It has important functions related to the brain, and it's actually known to interact with RNA-binding proteins, which from the work from Tammaryn's group we know is quite relevant for FTLD, and this includes TDP43. So OTUD4 is involved in stress granules, in RNA granules and stress granules, and these processes were already known to be dysregulated in these diseases. What we didn't know is that methylation was involved, so we were the first to describe that. We use a lot of bioinformatics tools and approaches, and one of them is creating networks, trying to understand how genes interact with each other and how they are related somehow. So we built co-methylation networks for the 3 data sets I was mentioning before, FTLD 1, 2, and 3. And we actually found signatures strongly associated with the disease, and that included that gene, AUTOD4. Because now we don't have just one gene, we have a signature. We can make sense of what these genes are doing. And so we can try to figure out what they have in common, what they are doing. We can also find out which type of cells are affected by dysregulation of these signatures. And across the data sets, we saw dysregulation of pyramidal neurons, which made some sense. But in one of the data sets, we also found dysregulation of oligodendrocytes, cell type that I mentioned before during my talk. And it's interesting that this FTLD3 dataset is composed by PSP cases, which are known to have tau pathology in oligodendrocytes, has coiled bodies. Continuing on the PSP front, we also found dysregulation of methylation in a gene called syntaxin 6. And this gene had been linked to the genetic risk of PSP before. So we wondered whether there was overlap between loci that are involved in genetic risk with loci that are involved in epigenetic risk, which seems to be the case for this gene. So Naomi, who started as a research assistant in my lab and is now doing her own PhD project, led this work where we looked at loci associated with genetic risk of FTLE, but trying to see if methylation could be dysregulating these loci. So in the case of syntaxin 6, we saw that the effect was not only seen in PSP, but was also seen in other subtypes of FTLD, including mutation carriers for MAPT, progranulin, and C9orf72. But we saw that across different loci, DNA methylation could indeed be influencing the dysregulation of those genes, even in the absence of certain genetic variants and mutations. We're moving on to Alzheimer's disease. So Kate, who is now a postdoctoral research fellow in my team, and also Hannah, who joined more recently, but did her master's between my lab and Dervis's lab, and she's now doing a PhD as well. So they looked into brain tissue, into methylation, gene expression, but we also complemented this work with transcriptomic data from mouse models of early stage Alzheimer's disease. What we found was dysregulation of an oligodendrocyte signature, and once again, as we had seen in MSA, this dysregulation was present across brain regions with different degrees of pathology. So, meaning that this is probably not just late-stage disease, but it's happening early. We also saw that this signature was shared with the mouse model. This mouse model doesn't have any tau pathology and represents early stages of amyloid pathology. And when we compare to other diseases, we could see the same dysregulation in other tauopathies, including PSP, so no amyloid pathology, and also alpha-synucleinopathies, such as MSA and PD. So this suggests that there's no need for amyloid pathology for this dysregulation to occur in oligodendrocytes, and it is suggesting that this oligodendrocyte dysfunction is happening early in Alzheimer's disease and perhaps other neurodegenerative diseases. Aging. Aging is a well-known major risk factor for everything related with neurodegeneration. So I have to talk about our work in this field. Although we all age chronologically in the same way, year by year, Based on our exposures during life, our biological age can be distorted in multiple ways. And because DNA methylation is at the interplay between the genetic makeup of an individual and their exposures, so we can use DNA methylation clocks to estimate this biological age. And so Meha did a study using data sets derived from human brain tissue and blood samples as well, including several subtypes of FTLD. And we indeed found accelerated aging in FTLD, and especially in PSP in the blood, to some degree in frontotemporal dementia as well. But in PSP, this effect, direction of effect, was consistent in the brain as well. And also studies from Meha have shown that the cells driving this age acceleration in the brain are not neurons, are not other glial cells, or the oligodendrocytes themselves. So they might be driving accelerated aging in the brain. I hope I have conveyed the message up to now that collaboration is key. And I'm also fortunate to be part of this initiative, the Deep Dementia Phenotyping DEMON Network, a global platform for innovation using data science and artificial intelligence that is led by David and Janice from the University of Exeter, but has members from all over the globe. And I'm fortunate to be part of the leadership team and to co-lead the Genetics and Omics Working Group. First with Nathan Skin from Imperial College, and more recently with Sarah from the NIH in the US. And coming back to connecting the dots. So our recent research have been showing that DNA methylation is playing an important role in dysregulating, driving dysregulation of gene expression in oligodendrocytes. And this is happening across diseases, neurodegenerative diseases. Oligodendrocyte genes and signatures are dysregulated across brain regions, some of them that almost don't show any pathology. It must mean that it's an early sign of the disease. These signatures are also shared with early stage mouse models of neurodegenerative diseases. And aging, which is a major risk factor for neurodegeneration, it seems that oligodendrocyte lineage is contributing to push the age towards older ages in the brain. So this raises the question, are oligodendrocytes a key missing link in neurodegeneration? We have been contributing to this. There are recent additional evidence towards this, but we will continue to work to be able to answer this question better. Because what we want is to work for a cure for Alzheimer's disease and other neurodegenerative diseases. Obviously, this work that I presented today was not just my work. It was the work of many people. In addition to everyone that I've acknowledged already during my talk, I would like to thank first to everyone that is part of or have passed through the Bettencourt lab in shorter or longer stays. Obviously, everyone, all patients and people that donated their samples, tissues, time, and their families, who make this, all of this possible. And a special thank to everyone that helped along the way. The lovely people from 1 Wakefield Street and everywhere at the IoN, Queen Square Brain Bank, the Lashley Lab, the EDI committee, and especially the Career Development Group with whom I've been working very closely with. Everyone that helped in many different ways. I could not include here everyone, and apologies if I forgot someone that I should have put the photo there. but they are all in my mind. If they know they contributed, I'm very thankful to everyone that helped along the way, and obviously the funders that made my research possible. Thank you very much.
Thank you, Sao, and good evening, everybody. So I have the pleasure of providing the vote of thanks for Sao. So I'd like to start off by thanking Sao for delivering a wonderful presentation and enlightening us on your journey to Queen Square. I've known Sao for many years as a friend and as a colleague, so it's an absolute pleasure and an honour for me to be asked to give this vote of thanks for your inaugural lecture. Having your family and friends here, both in the room and online, to celebrate us makes this occasion even more special. Tonight we've journeyed through the landscapes of genetics and epigenetics, guided by a scientist whose work has not only advanced our understanding of various diseases, but also illuminated the human stories behind the science. So your lecture was a masterclass in connecting the dots, from the Azorian origins that shaped your curiosity to the molecular signatures that define our neurological diseases. You've shown how rigorous science, deep compassion and relentless inquiry can come together to advance our understanding of the different diseases. Thank you for sharing your vision and your research, and especially your passion, with us. To the colleagues, collaborators and students that have walked along Sao for her journey, have contributed to this deep understanding, and to the patients and the families whose stories and samples have shaped Sao's research, your generosity are the foundation that everything we've heard tonight. Finally, thank you to each and every one of you for this evening, whether you're in person or joining remotely and being part of this celebration. Your presence affirms the importance of Sao's research and the community that it supports. I would like to extend my heartfelt thanks to Sao once again for delivering her professorial lecture. She should take immense pride in all her achievements in becoming a professor here at UCL, a recognition that is richly deserved. Let us leave tonight not only informed but inspired to keep asking questions and to keep seeking answers and to keep connecting those dots. Congratulations, Sao.
Right, it's a real privilege to introduce now the inaugural lecture of another exceptional female scientist. And obviously that's Jo. So Jo's journey combines clinical expertise with innovative science, and her work is really helping to shape the future of pediatric neurology and gene therapy. So she began her medical training in the University of Dundee, and then she went to do her specialist training at Sheffield Children's Hospital before completing her subspecialty training in pediatric neurology at UCL Great Ormond Street Hospital. So after she became a consultant pediatric neurologist, she was awarded a very highly competitive MRC doctoral fellowship to pursue a PhD in gene therapy here at UCL, focusing on dopamine transporter deficiency syndrome, a rare childhood neurological condition. So this really is what marked, I think, her transition into from the clinical world into the research. And just for the doctoral work, she received 2 prizes, the British Society for Cell and Gene Therapy's Fair Brain Award and the Otto Wolff Translational Neuroscience Award Prize. Saving the best for last, Jo finally decided to join the UCL Institute of Neurology in 2023, where she now leads the research group in the Gene Therapy Accelerator Center, and she also serves as the academic lead for the IoN DRI genetic therapy platform. Her work is really bridging clinical care, research and translation, but she's also a generous mentor, and she's a very collaborative colleague. So Jo is not only deeply talented, she's also an enabling and incredibly kind colleague. So I've also asked your research group, yeah, I can see you're scared now, Jo, of what they thought of you and whether they wanted to say a sentence or two. I allowed them to be a little bit cheeky. And here is what they say. Jo inspires us. She knows who she is and she knows what she wants. She leads with kindness, passion, and logic, even when managing the group feels like herding cats. Jo epically goes above and beyond to support us, always with grace and absolutely no hesitation in playing the bad cop, especially when there are figures and stats involved. So please join me in welcoming Professor Ang to her inaugural lecture.
Thank you, Helen, for that kind introduction. And whoever it was, I will speak to you later. So really, thank you all for coming. It's a real honor to share my journey with you today. So I work at the Genetic Therapy Accelerator Center, but I really think it's about gene therapists, amazing clinicians who are all inspired by the patients. My family are here, and it's pretty obvious that there's a whole load of people just walking in who I know very well. It's pretty obvious that I'm Chinese, I think, and my dad is a big fan of Confucius, so I'm going to start with Confucius. So we have two lives, and the second one begins when we realize we only have one. And that is probably a bit like me. I probably have had two lives. So this is an AI-generated picture of me. So I'm sorry, Mum, but my hair's a bit messy, even by AI. And what's weird is somehow they put on two lab coats. I have no idea why you would wear two lab coats, but I'm wearing two lab coats. But that probably reflects that I trained fully as a pediatric neurologist, and then I changed my mind and wanted to become a scientist. So I am two lab coats, Jo. But where should we begin? We should start with my name. So it's the worst surname, or the most challenging surname without any vowels. But Chinese people always start with their name and their family. So most of you actually don't know my name to start with. This is my name, I know, right? So in Chinese, this is my surname. So in most East Asian cultures, your family name comes first. Family comes first. So my surname in Cantonese is So no one will ever pronounce it that way. And certainly you would never call your lab that. So I'm going to stick to the lab being the GTAC. Okay. The second character is Zhong. And that means I'm the second child. So I have an older brother, and he's here today. So again, I have a family, and I have a sibling. And the final character is Yan, so my name is Zhongyan. See, none of you knew my name. Right. So I was born at the start of a new day. So that's what my last character means, the new day, a new beginning. So my family originates from southern China, from a place called Shundap or Shenda. And this is our ancestral temple. So this is where we pay respects to our ancestors. So that is our surname there. So for hundreds of years, our family will have gone to pay our respects to our ancestors here. And we still go there, not very often, but what I remember of going there is a beautiful garden. They have extraordinary dragon boat races, but mainly the food. So Shenda's food is so good, it's recognized by UNESCO. So if you're really into Cantonese food, that's where you need to go. Famous people from Shenda is actually Bruce Lee's family is from there. So he's a very famous martial artist from the same area. And actually what Shenda is really known for is incredible businessmen. So many Hong Kong tycoons originate from Shenda. So my family, this is my granddad, he moved when he was 18 to Hong Kong. So many people from Shenzhen moved to Hong Kong. So the family has been moved to Hong Kong for about 100 years. And he had 9 children. They had 9 children. And my dad is 7 of 9. So let's go on to my dad. This is my dad. He's there. And so in some circles, he actually needs absolutely no introduction. And it looks like he's winning an Oscar. He's not. It's not an Oscar. But we call it Daddy's Oscar. He won an award about 10 years ago called 中华之光, which means he is the brilliance of China. So just a minor title. So he's recognized very much for his promotion of education of Chinese language and culture all across the world, so in the UK and in Europe. So he has a close link with UCL, where he's worked very closely with Professor Li Wei, the director of the Institute of Education, for a very long time. So I met Li Wei when he was a PhD student. Actually, I'm not in his thesis, though. And these are my dad's awards. So this is his office, but I think actually probably the front row of this, the front of this auditorium also have trophy cabinets like this. I mean, you're all, highly revered. But this is his bookcase at home. And I love this bookcase. He also has an MBE here. Okay, he keeps that one at home. I don't know why. Is that at the top is the pictures of my brother and my graduation. So I think he would say that we are achievements as well. But I think my father's greatest achievement is that he married my mother, for sure. And so my mum is also from Hong Kong. Her family have been in Hong Kong for hundreds of years. And I was really lucky. So when I was little, till I was about 14, she was just my mum. That was her job. And she was amazing. I don't think we played together very much, but anyway, this was the first time I played cards. And I'd lost, and I was devastated. I cried and cried. And so Mum told me, well, it doesn't matter that you lost. Someone has to lose. It's about how you deal with it, and that you learn how to play again, and you don't have to be upset. Most people would probably just play again and let you win. But I was 5. She was already telling me how to think and think of other people. So with that, and so this is our family. So Mum, Dad, my brother, his wife, and my niece, who's taller than me now, and then my husband, Bryce. So very successful family, I would say. And if you stand next to my dad long enough, you might have to hold an award. Or you might receive one, actually. So the Chinese government actually asked me to apply for a Future Leader Award, which they did give me on merit last year. I think the most significant thing my dad did for me is he moved the family to Newcastle, so I'm a Geordie. We became Geordies. I love Newcastle, though. So this is the Tyne Bridge, and I love the Great North Run as well. I went to a very academic girls' school, and what I really liked was playing piano. So this was my first piano recital when I was about 8. And that's kind of what I really like doing. This is probably my last piano recital anyway. So I loved my music teacher. I loved being in choir, and I was really lucky that with choir we were able to perform at the Royal Albert Hall. So that was great, but it's probably the last time I sang as well. And this is my 6th form class. So the uniform was so terrible. I was so relieved to get to 6th form. So the uniform, I can't even show you. It was brown, and I have never worn brown since. So this is my form class, but there was probably 21 girls doing A-level chemistry. 18 of us went to medical school. 2 went to veterinary school, and then 1 became a dentist, of course. And Famida Chowdhury and I were in the same year, so she's a consultant in epilepsy at the National now. So very clever class, I think. So we became Geordies. And why did we do medicine? So there are several doctors in the family, but not my immediate family. But my brother went to medical school. So this is my brother. He's 4.5 years older than me. I don't know how I got so chubby, but anyway. And he went to the University of Dundee. And that has now become our family medical school. And he embraced Scottish life and is wearing a very good-looking kilt. And he is an NHS hero. He is an A&E consultant, leading an emergency department of 200 staff in the northeast. And what I learned really from my family and living in the north is you need to work proper hard and be extra canny to people. So canny means to be nice and to be good. So I went to Dundee, and at that time, Sir Alfred Cuschieri was there. So he pioneered laparoscopic surgery. So whenever he operated, all his operations were then shown around the medical school. You had to go watch them. And then also taught by Sir David Lane, who's a Nobel laureate who discovered P53. And so I graduated and looked really young. And this is my best friend, Quiza, who's here. She's a pediatric allergist in Cambridge now. And this is our other best friend, Seema, who's a GP in Australia. But perhaps this is the first pediatric neurologist I met. So that's Zoela Zol-Kipley, who is a neurologist in the States now, but she is Quiza's big sister. So maybe that's how I got interested in pediatric neurology. So I left knowing that I was interested in neurology and I was interested in pediatrics. And I got my first SHO rotation at Sheffield Children's Hospital. So it was a 3-year rotation, which was quite long, but incredible, because I got to do pediatrics, neonatology, pediatric neurology, and pediatric surgery. So my first publications, I have no idea why. Well, I do know why. I cannot tell you anything about them now. We're all in pediatric surgery. And you just wrote them just like that. Anyway, so 3 publications in 6 months. Actually, they always had the best trainees. So Jack Bartram here became a consultant at Great Ormond Street and has just moved to Sick Kids Toronto. What would happen really is that I would marry one of the co-authors a few years later. So my husband Bryce is here. He's a co-author. So you don't need to be on the apps or anything. Just write up some papers with people, maybe. But I think this was really fundamental, is that I got to do pediatric neurology with 3 amazing pediatric neurologists, Peter Baxter, Chris Ritty, and Santosh Mordekar. Peter said he was coming. I hope he made it. Anyway. And like my other job, I wrote some papers, just like that, in 6 months. I was so lucky that I had a pediatric neurologist allocated to be my educational supervisor from very early on. But I had a real problem. We used to have meet and have our educational supervision, and you can see these things that you need to fill in. And there's this section on careers advice, and I cannot for the life of me read it. So if someone can tell me what he wrote, I'll try and do it now. Okay. Is it just go to Queen Square? Okay. But Peter was very generous. He's a brilliant mind and always had really interesting questions about how we can standardize clinical assessments, because it's really difficult in children. So what's normal for muscle strength in a small child is completely different to an adolescent, for example. So he set about with this project to try and standardise different kind of motor assessments as proxy measures for muscle strength. So you can get a child to stand up from supine or see how quickly they walk or run 10 metres. He wanted me to generate some centile charts. So I said, okay, let's do it then. And I have no idea how I did it, because I was a full-time clinical trainee, but somehow we recruited 320 children from nurseries and schools across Sheffield, and I would go out, I don't know when I had the free time to do it, to video and time all these children. And then we worked with the statisticians from DevMed Child Neurology, so Peter was the editor at the time, to generate these centile charts. But I really don't know how I did it. But it was an amazing experience and my first real feel of research. And, you know, Peter is highly revered, became the president of the BPNA, and he edited this book on neurometabolic conditions, the vitamin-responsive conditions in paediatric neurology. And so perhaps that is how I started to become interested in neurometabolic disorders. And then really with Peter's support, I really wanted to pursue training in a centre of the top excellence. So I moved to Great Ormond Street. And somehow I have worked at Great Ormond Street Paediatric Neurology as an SHO, an SPR and a consultant. I don't know if anyone's done that. I don't know what I'll be if I work there again. But anyway, and the department, the favourite time of the department is probably when I was an SHO. And it was quite relatively small compared to now. And so we had the chair of epilepsy was Brian Neville, and now you probably know Helen Cross. Rod Scott was there. He actually gave the departmental seminar recently, but he's now in the States. I know Sarah and Das. But my true love was acute neurology. And I was fortunate to be able to work with Professor Robert Surtees. And then the acute kind of neurology team was Carlos de Souza, Lucinda Carr, Catherine DeVille, Jay Gleason, and Shell Hemingway and Sarah Benton, who's passed away. And these were the registrars. And these registrars are now professors all over the world, all leading the department now. And the board was relatively small, but we somehow knew what was going on with the patients and we communicated really well. So to me, this is like a forgotten age, almost. And thank God I got a different educational supervisor whose handwriting I could read. So this is from Catherine DeVille, who's here, and it's also her birthday today. So happy birthday, Catherine. And so then I moved on to do my registrar training, and they sent me to do Witz Neuromuscular Centre. And I didn't really know much about muscle, to be honest. And mainly I made friends. And they're still my friends to date. So Reagan Foley is an American neuromuscular expert at the NIH now, and she had moved over to do an MD with Francesco Muntoni. So she was already a neuromuscular expert. And at that time, she was working on collagen 6, but in her spare time doing gene discovery, because you do gene discovery in your spare time. And she was characterizing patients with Brown-Violetto-Van Leer syndrome. And I said, Peter, we looked after a patient of Peter's like this for many years at Sheffield. Please contact him. And indeed, she diagnosed the patient again with riboflavin transporter deficiency. So Reagan was a reader at our wedding, and I still see her now. So this was probably last year. And then another great friend, and I think they're all online actually, was Anne Matthews. So she was just a workhorse, could do everything. And now she is a leading neuromuscular specialist in India and has, I don't know, thousands of patients. So she says to me, actually, she was so clinical, she didn't do any research while she was in London. Could I help? And I was like, well, I'm not sure. So she sent her fellow my way, and every few weeks I would help them look through their clinical data on SMA patients. So we've got a small paper out recently, and also looked at how they've dosed 35 SMA children with Zolgensma, which they've been able to obtain through compassionate use or for developing world access. So I'm still working with them now. So in neuromuscular, I made friends. I did not become a neuromuscular expert. And then in acute neurology, I think that was my true love, and I did some work on Moya-Moya syndrome, which is where you get recurrent strokes and a cerebral arteriopathy. And I just went through about 70 patients with Moya-Moya disease who had undergone neurosurgical revascularization surgery and compared two surgical techniques, looking at direct bypass or ECIC, where you do multiple burr holes. And there was no significant difference between the techniques, but they appear to improve the risk of stroke recurrence. So I was doing all of this in my spare time. I really don't know how I did it. And I thought, actually, I really wanted to do neurometabolic, to be honest, but it was difficult because Robert Surtees had passed away. And I really wanted to find a way to do formal research. So I then ended up becoming a consultant, essentially. But what I'm really known for is developing gene therapy for dopamine transporter deficiency syndrome. And this also links back to Robert Surtees and a bit of my time as a registrar. So in about 2004, Robert and Georg Hoffman had described 4 patients with infantile Parkinsonism dystonia. And they had these really strange neurotransmitter profiles where they had high dopamine metabolites, but you think people with Parkinsonian symptoms should have low dopamine metabolites. And then when I was back in Sheffield as a registrar, Santosh Mordekar had identified 2 children with cerebral palsy in the same family, which was pretty unusual to have 2 cousins with cerebral palsy. And we also identified 2 children in the same kindred with infantile Parkinson's dystonia, and they had the raised CSF dopamine metabolites. So we had spoken to Robert and Georg Hoffman, and I'm a real nerd, so I got him to sign my book. We were going to kind of move things forwards towards trying to diagnose these patients. But sadly, Robert passed away at almost the same time. So he has a bench in Queen Square, and he really was a very dedicated clinician, an inspired teacher, and a true genius of his generation. He really was. So he was a big loss to the department and to the field. But then the genetic revolution came along, and Manju came along and really changed my life. So everything changed when Manju came. So Manju was doing her PhD in Birmingham and then moved to Great Ormond Street and the Institute of Child Health as a consultant. And she had just identified the causative gene for dopamine transporter deficiency syndrome. And I'm still reading this paper, I don't know, every year. Anyway, so she had identified that children with this infantile Parkinsonism dystonia and high dopamine metabolites had dopamine transporter deficiency. So it's biallelic loss of function mutations in the SLC6A3 gene. And they have a raised homovalinic acid to HIA ratio. And at the moment, there are about 28 published cases, but now I think we've expanded that out to over 50. And she really defined the clinical phenotype, where you start off with this hyperkinetic movement disorder in infancy, and then it progresses to severe childhood Parkinsonism before the age of 10. So you can see this video is playing where the child is incredibly bradykinetic and rigid. And so what does a dopamine transporter do? So I've spent years thinking about just being in this diagram. So dopamine neurotransmission, many of you may know, is that you convert tyrosine to levodopa with TH, tyrosine hydroxylase, levodopa to dopamine with AADC. Dopamine is packaged into vesicles by VMAT2, vesicular monoamine transporter, and then released across the synaptic cleft to stimulate dopamine 2 receptors. The transporter then terminates neurotransmission for dopamine reuptake for recycling of dopamine, or dopamine is catabolized by MAO or COMT to homovalenic acid. So when there's complete loss of function of the dopamine transporter, there's failed dopamine homeostasis, and it's kind of sitting around in the synaptic cleft, and then is catabolized. So you get this kind of aberrant HVA, which is raised with normal HIA, but these Parkinsonian symptoms. So Manju changed my life, and with her support, I got an MRC fellowship to do a gene therapy PhD in dopamine transporter deficiency syndrome. And so one of the first things we were looking at is a new patient cohort. And what we identified was a really atypical clinical presentation, where these were a single kindred of 3 brothers, where they presented with a head tremor. And then this is the older brother, where the disease has really progressed. And he's more frankly Parkinsonian with head tremor, ocular flutter, he's bradykinetic and has postural instability as well. So this is really different to the infantile patients that were characterized initially. And we did some in vitro studies to model the missense variant identified in this kindred, and identified that they had higher residual dopamine uptake, so they had retained some function, whilst all the infantile onset probably had less than 5% dopamine transporter function. So there seems to be a genotype-phenotype correlation, where the more transporter function you retain, the later the onset of disease. And this was corroborated by a Danish group as well, where there's an adult who presented at 28 with unilateral tremor. So you can see he has early onset PD, and he had on his in vitro studies about 33% dopamine transporter function. So we then understood that if we could restore some dopamine transporter function, that we likely could have quite a significant therapeutic impact. So even moving from 5 to 8, 9% would improve motor function significantly. So we had therefore hypothesized that gene supplementation with using an adeno-associated virus could be therapeutic. So for those of you who don't know about this, most of you do, is that you have a gene therapy construct. Essentially, you have viral packaging sequences, a promoter for transcriptional regulation, and you express a transgene, and you package it into an AAV capsid. You deliver it, and it's uptaken by different receptors, goes through the endosome and Golgi to enter the nuclear pore, where the capsid uncoats to release single-stranded DNA or self-complementary. And then there's second-strand synthesis and a transcription translation to express your therapeutic protein. And what was happening in the field at the time was actually a gene therapy development for a very similar disease, for ADC deficiency. So the enzyme that converts levodopa to dopamine. So this is another ultra-rare neurotransmitter disorder where you get biallelic loss of functions in the DDC gene. It's ultra-rare, but there's a founder mutation in East Asians, so Taiwanese population in particular. And these patients have no dopamine or serotonin, and they don't respond to L-DOPA, and they have reduced survival. So this is a child with ADC, so very severe with very little movement, and they have significant problems with ocular gyrate crises as well. So they had developed an ADC gene therapy for Parkinson's disease, and these families said, could we try it for ADC deficiencies? And I think these families are incredibly brave. And their clinician, who's a geneticist, was incredibly brave. So they were able to use this on in a compassionate trial in Taiwan, so in 2012. And this was the start of ADC gene therapy. And this is a post-treatment child where you can see that they're just so much better. There's no way an untreated ADC child would be able to do this. So what they developed was bilateral putaminal infusions into the brain of ADC gene therapy to restore ADC function. So in various clinical trials, they have dosed over 30 patients in several phase 1 to 3 trials, and this received international regulatory approval in 2023 and is now available on the NHS. So they are dosing patients at Great Ormond Street now, and it's amazing. Manju should tell you about it. So then we thought, well, dopamine transporter is a similar disease. It's at the bottom of this pathway, and we could develop a translatable gene therapy as well. So this is work, and really the main chunk of my MRC fellowship with Manju Korean and Simon Waddington, who's a gene therapist at UCL, and also with Serena Barrell, who was a postdoc in Manju's lab at the time. So Serena led essentially, so there was 2 prongs to this. One was that we would develop iPSC and a dopaminergic neuronal line to evaluate gene therapy in vitro. That was led by Manju's lab and Serena, and that Simon and I would do the in vivo bit, the gene therapy in vivo bit, and we're trying to get this back to the patients now. So this was published in Science Translational Medicine, and the first part, this is Serena's work, is that we tested a lentiviral construct in the dopaminergic neuronal model. So what we could see is that we have 2 patient lines, and that they were transduced with a GFP lentivirus and a lentivirus with dopamine transporter and GFP. And essentially there is significantly improved survival and increased tyrosine hydroxylase positive neurons. So we could see that there was better survival following gene therapy. And then we did a dopamine uptake assay and significantly improved dopamine transporter function. So there was proof of principle in our patient-derived neuronal model that gene supplementation would restore neuronal survival and dopamine transporter function. And so we then wanted to move this in vivo. So we got the dopamine transporter knockout mouse, which initially is hyperkinetic. So we've got a knockout mouse running around showing stigmataxis behavior. And its wild-type littermates who were just totally not bothered. And then at 5 weeks, they suddenly become sick and actually bradykinetic and lose weight. So actually similar to the patient phenotype I was showing before. We gave the mice gene therapy at birth into the ventricle, and this is the normal survival, about 40% untreated. And with gene therapy, it was restored to 100%. And there was a significant improvement in body weight as well. So we were shared this mouse from Mark Caron from Duke University, and like a real nerd, I got him to sign his paper, and it's framed in my office. So he said to Joe, best of luck with a DAP knockout mouse. And he said, I made him feel like Katy Perry. So we did lots of neurobehavioral and locomotor studies, and we did them for a whole year because I wanted to see if the mice would become Parkinsonian despite gene therapy, and actually we restored their motor function for a whole year. So this is a treated mouse who is quite big and just exploring normally, and we've got this hyperkinetic anxious mouse which is untreated. We did neurotransmitter analysis with Simon Heels and Simon Pope. We were able to modify the neurotransmitter metabolites, and also Gabri did electrophysiology for us on dorsal striatal MSMs. So in wild-type mice, you have a bimodal firing pattern. In the knockouts with the high dopamine, it's all high firing, and then following gene therapy, we restored it to bimodal. So we had really good data. But we had a problem, because there's always problems, is that with the AV9 gene therapy, we had achieved expression throughout the brain. So we're certainly expressing it in the target regions, but also in the off-target regions. And in the mouse model, we were seeing an astroglial response in the frontal cortex. So we had to really think about how best to make this a clinically applicable gene therapy. So we got MRC funding for my postdoctoral position, where we did adult stereotactic delivery of AAV gene therapy and changed the capsid to an AAV2, which restricts the expression really to the region of infusion much more. So then I had to learn midbrain stereotactic injections and did a dose ranging study. And we saw a dose response with survival, where the lowest dose had some effect, but middle and high doses had 100% survival. There was no effect on body weight when treating the mice at an older age. And we again restored the motor behavior on open field. And we had negated the problem where the off-target is no longer causing an astroglial response. So this was quite a journey for gene therapy for dopamine transporter, where Manju identified the causative gene and characterized the clinical phenotype. A few years later, during my PhD, we identified atypical patients. Serena showed the proof of principle in the lentiviral treatment of the iPS-derived neurons and the AAV9 gene therapy in neonatal mice. But really, to move this towards the clinic, we did AAV2 midbrain stereotactic gene therapy to minimize off-target effects. So since then, we've received orphan drug designation internationally and attested scale-up vector manufacture. We've really developed a big patient cohort now, and it's with Manju towards clinical trial, basically, which is amazing. But obviously, it wasn't just me and Manju. There was a huge part played by Simon. So this is Simon Waddington, who leads the Gene Transfer Technology Group, where I did all my in vivo work and gene therapy. And he literally flies around the world injecting mice or primates or whatever to test different gene therapies. And he's an amazing scientist and gene therapist. Apparently, I was his first PhD student. I'm not sure how. And this is me winning the Fairburn Award from the British Gene Therapy Society. But Simon has been a wonderful mentor for many gene therapists across UCL, London, probably nationally and internationally. But these were probably my contemporaries, some of whom you may know. And so it was really no surprise that he would mentor us all to become independent gene therapists. Like Ahad Rahim, Rajvinder Kada, John Council, William Baratow, and Natalie Suffand, Ashley at King's. So it was Simon who encouraged me to move to IoN. And I missed them terribly. But we were lucky that actually my PhD student, Ricardo, would come and join us. And then another PhD student, Constantina, from the Institute would also come to join us. So I tried to get the band back together, as it were. And so I joined the Genetic Therapy Accelerator Centre. And the aim of GTAC is to embed RNA and AAV genetic therapies in basic and clinical research. And it's an initiative across IoN, ICH, with the support of Mike Hanna and Helen Cross. So Francesco is our director, and Sarah Guti is a senior fellow in RNA therapies. And so they said they wanted an AAV person, and they thought it might be me. And so there aren't many people who have worked with me, both as a clinician and then as a scientist. And so when you work in the Dubovitz Neuromuscular Center and you finish, your thanks is Victor Dubovitz. It's an autobiography, and everyone signs it. I had never opened this book. until yesterday. But there's a really nice note from Adnan, and who I think I worked most closely with when I was working with in neuromuscular. This is Francesco's picture in the book. So at the Hammersmith, he was black and white. But apparently this is what he wrote. So I would say easier to read than Peter, but nothing like Catherine's handwriting. Anyway. And almost, I think maybe on the first or second day, I ran into Simon Heels and Simon Pope in the lift. And they said, "What are you doing here?" I was like, "Well, yes, exactly." So I'm going to open an AV lab, I think. And where are you? We're on the 9th floor in lab 924. And they were like, "Oh, we used to be in 924. And I was like, "Really?" "Yeah, it used to be the neurotransmitter lab." This is the lab where they ran the first CSF samples, where they first discovered tyrosine hydroxylase deficiency, AADC deficiency, and dopamine transporter deficiency. So it's the lab where they identify the biomarkers for the diseases that I now try to cure. So I don't know how serendipitous that is. This is a picture of the AADC Trust, which is led by Lisa Flint, whose child is affected. This is Simon Heels, Peter Clayton, and Robert Surtees, who discovered the disorder and causative gene. And they're in the foyer seminar room, and this is where I have my lab meeting now. So I sort of feel we've come full circle, really. So we moved to Genetic Therapy Accelerator Centre in 2023, so a little over 2 years ago. I came with everything I had, which is not a lot. Okay, so I had 3 boxes, but everything I had also was an amazing RA, Hester, who is a PhD student as well. And in the first year at the Institute, I just seemed to run a lot. So I ran the London Marathon for Great Ormond Street. I did an ultra marathon, so I ran 50 miles with my sister-in-law just to keep her company for no good reason. And I did the Great North Run because I love going back to Newcastle. And I'm slightly hysterical here because I'd run a half marathon and then it poured on us, so we were freezing. But what I really came to the Institute with was a huge amount of translational research experience and support. And being successful in getting translational grants. And that's really working very closely with the Translational Research Office for a long time, so since my postdoc. And I think I was just so well trained by Joanne, who was at the TRO at the time, when we were getting our MRC grants, and also had a relationship with Katrina Crombie at that time, who was at the MRC. So they're both at LifeArc. And now I continue to work with the TRO. So Celia manages our 2 LifeArc grants. Helene has helped us with TAS awards and MRC grants. And Jane recently sent me to Slovenia, where we've developed a protein engineering collaboration with Roman as well. And Katrina from UCL Business has really helped us license our gene therapies to industry and help us with patent applications. So I came to the Institute with all this support and thankfully a project grant from LifeArc to get started straight away. So this is gene therapy for another infantile Parkinsonism. So we have gene therapy for AADC already. We work downstream with dopamine transporter, so we've gone up. So we're working on TH. This project is 2.5 years in, led by Hester and Rebecca, postdoc in my group. And the data is amazing, but I'm not going to show you any of it. I'm going to let Hester and Rebecca share this data. So they presented it at the departmental research meeting last week. We're really fortunate to get the mouse model from Aurora Martinez and Beattoni in Bergen and Zurich. And again, we get to work with Simon Heels and Simon Pope on neurotransmitter analysis. And so it's at late preclinical stages where we're now really going to meet with the regulators next week, I think. And we've been speaking with the International Working Group on Neurotransmitter-related disorders about natural history study, patient family groups as well. So this is really moving forwards towards clinical application as well. I said I was getting the band back together. So Ricardo was our PhD student with Simon at the Institute for Women's Health. And he did a project with AskBio developing novel promoters. So he has developed a bioinformatics platform to generate hybrid promoters for CNS activity. And he's bringing some of that work back to the lab now. And he won the American Gene Therapy Society Meritorious Award for this work. And then this year we got a big boost in our funding to develop gene therapy for monogenic Parkinson's, so PARC19. So during my PhD, we diagnosed some patients with DNA JC6 juvenile Parkinsonism dystonia. And Manju's lab developed an IPS neuronal model. We were able, again, to show proof of concept with lentiviral gene therapy that you could restore synaptic function in these neurons. So we were able to get the mouse model from Patrick Lewis and Mark Cookson. And we got a small grant initially from the TRO and Rose Trees to appoint Camilla, who's our RA PhD student, to do some initial work on gene therapy for PARC19. And based on that, we've been able to get over $1 million to develop this project even further. So this has just started. I also lead the genetic therapy platform, which is an AAV packaging service at the minute. So I've sat on a committee with very important people for about 2 years, planning the genetic therapy platform to support AAV research and AAV production for researchers across the institute, and working with JP Schiavo, David Howard and Lee Stanier from the strategy team about equipment and logistics with the delay. So we have now set up the genetic therapy platform to support AAV packaging at Queen Square House. So we've packaged our first AAVs for the Institute in April. We've done 8 so far. And the work at the moment is being absorbed by the research group, and mainly supported by Rebecca, to which I give many thanks. We also invested in this amazing machine at AAV capsid analysis. And really we need to better understand how we analyze our AAVs and the quality of our AAVs. So with this refined mass photometer, we're able to look at the empty-full capsid ratios. And now we have colleagues from all over UCL, so from School of Pharmacy, Bioengineering, Hematology, coming to use our machine to analyze their AAVs. So what else do we do? I also support gene therapy projects across IoN. So I've somehow collected 3 new PhD students from different groups. So gene therapy in leukodystrophy, a periodic paralysis novel therapies project, and a mitochondrial disease gene therapy from Henry Holden, Viorica Chelban, Rupi is here, and Rob Pick-Heasley from leading the Mitochondrial Centre. So we're quite busy. And then there's a couple more where we're working on paediatric neurodegeneration with Dr. Barrell and new postdoc Francesco. And we've just started a new project today on genetic ataxia with Selena Ray as well. So we're pretty busy. And of course, you can't work with Francesco Muntoni without doing neuromuscular disease. So there are 4 projects in AV for neuromuscular disorders as well, with lots of different people. And so I'm coming to an end, and really would like to thank the department. So we're led by 2 leads, Nick Fox and Sarah Tabrizzi, but I really must thank Sarah and Helene for their support of my promotion. I was very surprised, to be honest. And we've not been at IoN or the department for very long, but what has actually been a great help for me is chairing the departmental seminar series. So I really gotten to know the department very well, and the research fellows and PhD students, and their presentations are always phenomenal. I mean, they're just amazing. I know so much about Huntington from Ed and the department that I was on the CNS abstract committee for the American Gene Therapy Society this year, that I selected Huntington gene therapy oral presentations. And when I chaired the meeting, I was asking them about MSH and FAN1 and almost forgot to ask them about their new capsids. And the other co-chair was like, they're capsid. Anyway, so I now know a lot about Huntington. I'd like to thank Selena, who got me involved in organising the UCL Neuroscience Symposium. And just in good faith, I can say I ran a half marathon for ARUK as well. We have the most amazing administrative support in D&D, so we must thank Sophie, who supports the Pan-UCL AV Symposium that we've been running for 2 years, and she supports some of the work with the Vector Core as well. And in particular, the administrators, Rosie, who was here when I started but recently retired, but her wisdom and guidance were invaluable, to be honest, so she kept me safe. And now we have Sabrina, who is also amazing, and I'm sorry for all the emails. And Sue and Mark, who've helped us orientate into the lab. Sue is amazing. I essentially ordered the biggest hood on the planet. It's bigger than my office, but somehow she managed to get it into our lab. I think I can lie down in our hood anyway. And in the main, thank you to the group, thank you to GTAC. You are amazing. I know I ask a lot of you and you work really very hard, but we've done a lot, I would say. Thank you to Francesco, who has been a great support and really made us feel part of the Institute and also part of ICH and his group there, and to our funders. And really, thank you for all your attention. And last but not least, is there are people, certainly online, but there are people who really couldn't be here. So my grandfather passed away 8 years ago, and my father-in-law passed away last year. My mother-in-law lives in Goa, so I was able to celebrate with her in Goa a couple of weeks ago. And my grandmother lives in Hong Kong, and again, we had a celebratory dinner together as well a couple of weeks ago. But last but not least, to thank Bryce, who is an amazing paediatric surgeon in Dublin and has many achievements of his own. So he's an associate professor at UCD. He is utterly selfless and allows me to be here. He taught me how to suture for the stereotactic surgery. So this is me practicing on nectarines at home. And so I know our mice have the best scars and the best surgical tools in UCL. And really, I know I should be in Ireland a lot more. Thank you, everyone.
I didn't expect to learn so many things about you. So I thank you on my behalf of everybody who works with you, for also putting a personal touch. We learn a lot of things about. You need to give me a tutorial on how to pronounce your name properly. And I think your scientific part of the presentation suggests to me that you will do a gender balance of the famous people from Shoot. I only saw some males there. I think there will be a female there, and that will be you. I think that as you indicated, you are, to some extent, yes, you work and study in other places, but most of your work has been done here, and I met you during the pediatric neurology neuromuscular training. thank you for putting the handwritten note to say we'll take you back if you wish. That was good foresight. I think that your scientific output, I think UCL has appropriately quantified and decided that they are outstanding. You won't get a chair at your position at UCL without having done that. But I think what you have been also able to do it, you are really a column, you are a support for your own team, and I think I've seen how your team loves you and how much they are. You push them, and I think what you mentioned regarding the genetic therapy accelerator now and the vector core providing vector for UCL at the moment supported by your team. So it's a testimony to you, to your team. But I think what you do it, and you're able to manage people well, this is not by bullying people, but by having everybody working together and recognizing the contribution to all colleagues. So that looks something that should happen all the time. It doesn't. So I think thank you for that. I think that perhaps I would, if you like, your sensitivity and respect for the others, therefore, have come through your presentation, and that's clear. I would like to say two more things regarding UCL as well. In 2021, 2022, I convinced Mike, Alan Thompson, and the faculty from Ibrahim, and the original director at ICH was just before Helene, that it was timely to appoint, to establish a center to take forward the RNA and AAV gene therapy. And I think that the investment, therefore, was part of the vision of the faculties, the two directors of the faculties, and the two dean of the faculty, and to the directors. And I think you probably already repaid back all the investment and more with your own grants. So I think it just shows two things. Firstly, that considering that I put the business case probably at the early 2022, that at UCL things can also move very fast, and I think that was fantastic. I think that you mentioned towards the end, and that in my second thing, the support that you have received, but we all have received in the Department of Neurodegenerative Disease, I think has been second to none. And I think your promotion was very, very strongly recommended by Sarah, by GP, and other colleagues who felt your achievement would really benefit from this. And I think for me, putting together your success, the fact that the faculties have seen the need to establish this particular genetic therapy accelerator, and the fact that UCL and the Department of Neurodegenerative Disorder, IoN, have quite rapidly, you've only been appointed 2 years ago, have been able to agree on your promotion. I think altogether a sense that a good person who works in a good institute, in a good university that recognize priorities, and in a good institute that recognize talent, I think a good way to finish this speech. Thank you very much.